So thank you very much for joining us today. Great turnout. I know that it's always time away from patients, so we really appreciate it. So today is a really exciting workshop for us because it's something we've never done before. We've never done a brain workshop before. It's the first one, so it's not been done in Johannesburg or Durban. And the one thing you'll also notice is we're changing the format of our workshops. And the most exciting thing for me is actually that I'm like the introductory speaker. And I don't get to talk for 4 hours. So we have some wonderful speakers following me today who bring amazing knowledge and insights into the program. And I think you'll appreciate as we go on how we're looking to do things differently in the future with really different speakers coming in to share. So I will introduce them as we go along. But essentially, I'm going to start. I've kind of been given the task of really bringing the subject into the room and hopefully getting your attention. So what's 50 million? Anyone want to guess what's 50 million?
What I'd like to win in the lottery.
Me too. Okay, that's not what it is. It's actually people with Alzheimer's in the world today. 50 million. 1 in 6 of us will get Alzheimer's. So sitting in the room alongside you in pretty much in rows of 6, 1 in 6 will go on to get Alzheimer's in our lifetime. Cognitive decline and Alzheimer's and all those kind of associated conditions is the 3rd leading cause of mortality in the world following cardiovascular disease and cancer. The 3rd. I mean, that's just extraordinary. It's like, where did it suddenly arrive from? And yet we're finding that it's now right up there. And as we say, everyone knows someone who's a cancer survivor, but no one knows an Alzheimer's survivor. So in that way, what are we actually doing wrong when we have a 3rd leading cause and we have no survivors? Another statistic, this one's for Julie. 75 million Americans have a badly spelled heterozygote, my apologies. So 75 million Americans have ApoE4 1 allele. 7 million Americans have E4E4. That's a lot more common than we consider. Unfortunately, I don't have South African stats, but I could try work them out. 0 cures. As things stand right now, we do not have a cure for cognitive decline or Alzheimer's. This is a Nobel laureate from the early 1900s. And what is striking about his commentary is he said that the nerve paths are something fixed and immutable. Everything must die, nothing may be regenerated. Quite a frightening comment, but this is really where the science was sitting in the early 1900s. We move forward to Dr. Peter Eriksson, who was a Swedish scientist who did some amazing neuroscience. He was a neuroscientist. He said that his study demonstrated that cell genesis occurs in human brains and that the human brain retains the potential for self-renewal throughout life. And I think that's the most important, is the last part. Self-renewal throughout life. So really we've come an extraordinary amount of way from early 1900s to the late 1900s. Okay. The reason I have the slide is that last year, May 2017, I went to the IFM conference in Los Angeles, this one over here. And the theme for the conference was the dynamic brain. I was very lucky to be there. It's essentially 3 days of back-to-back lectures all about the brain. And what was even better is that they gave me all the PowerPoint slides and they gave us all the audio lectures as well. So really, there's no ways you can take it in 3 days. So it was really amazing to be able to have this resource to dip into again. So what I did when I was preparing for today was thinking, what I really want to bring to you is a sense of what's happening out there in terms of brain health. So not just this is ApoE or this is MTHFR, but really what is happening in the world of brain health that is changing the paradigm or changing the environment in which we practice. Because if we come in there and say, we can't do anything about it, there's no reversal, there's no cure, there's no way of treating, we're kind of going back to the early 1900s. There are 4 of the speakers that, in my opinion, are the real thought leaders in the space of brain health. All the speakers were fantastic, but these are the 4 that are most recognized for their work in brain health. Joseph Pizzorno, whose speciality, actually they're wrong. Thanks, Jason. But anyway, Joseph Pizzorno on the right with the gray hair, okay. Dale Breston on the left with the Botox, okay. David Palmetta is correct, and Tanzi is the Italian-looking chap with the very dark hair. And really, they all bring something quite unique to the conversation around brain health. And today, I'm going to bring you some of their thinking, taken from the best of them, about really what they think is happening in our field. So what is happening in brain health? And these are some of the key topics I'm going to cover. I wasn't given a lot of time to talk, so I'm going to really try and move through it quite quickly. But hopefully we'll just give you some insight. Okay, the first thing we're going to discuss is BDNF, exercise, neuroinflammation, toxic exposure, infection, gut-brain connection, and of course, my beloved genetics right at the end. So let's talk about BDNF. So some of you might be familiar. How many of you are familiar with BDNF? You should start, it's coming through in the literature a great deal. You should recognize it from the new genetic test. BDNF is in there, one of the variants. But it really is one of the hottest biomarkers or the hottest kind of measures of brain function. It's basically a protein that is produced inside nerve cells, particularly. And we call it, the easiest way to remember BDNF is the fertilizer for the brain. The more BDNF we have, the better the brain works. So if you only look at the 2 bottom sentences, that is the most important thing to remember. Higher BDNF, better brain function. Fertilizer for the brain. The more we have it, the better our brain functions. Just to give you some insight, when BDNF is lower, well, we should say when these conditions are present, BDNF is always lower. So it's one of the biomarkers that we use to measure brain function or how well we're doing with our brain, okay? What does BDNF do? Just going to touch on it very, very quickly. What does BDNF do? So just a few key issues, just a few things it does. Neurogenesis, neural differentiation, appetite, telomerase, development, maintenance, and function of the CNS, long-term potentiation in hippocampus. So they've actually shown that levels of BDNF can be correlated to the size of the hippocampus and blocks injury-related apoptosis. In other words, you can see that BDNF is an absolute essential biomarker of brain function. Right, as I say, I don't get to talk about any one thing very, very long, so I'm going to have to move on. One of the most exciting things that I encountered at the IFM conference on the dynamic brain, that was my major take-home message, was around exercise. I was absolutely blown away by the power of exercise in terms of cognitive brain function. And I remember one comment, I think it was from Tanzi, who said, if we do nothing, we have to do exercise. And of course, the most important exercise is your right-left exercise. And this is what I took home. There is a 50% reduction in cognitive loss when we exercise. And there are so many papers written on this, so much discussion. I mean, it's been beautifully shown. So, I mean, this paper in itself is outstanding. It's open access, and if you want me to send it to you, I can with pleasure. But it basically explains what is the process of exercise that drives brain function and neurogenesis so much. So this is a topic that could go for hours. Unfortunately, I can't. The other thing is exercise is the most powerful variable in terms of increasing BDNF by far. Okay, neuroinflammation. So we're all very comfortable with inflammation. We're all familiar with inflammation. We know that that's a root cause of chronic disease. It's one of the reasons why it sits right up high in cellular function in our genetic report. But in brain, it's absolutely fundamental. So I was asked the question yesterday, why didn't I put all the inflammatory genes into the brain health panel? Excellent question. The reality is there's so many genes in inflammation that if I put it into the brain health panel, it would have diluted all the other genes. So whenever I look at brain health as an issue, I always look at inflammation and brain health together and put them together, because it's such an important pathway in itself that needs to be considered. So it's described really as this kind of seesaw, this real fulcrum, that on one side you have inflammation, and of course I'm talking about low-grade chronic inflammation, and on the other side you have neurogenesis, building new brain cells, growing synapses, all of that. The more inflammation we have, as the inflammation seesaw goes up, our neurogenesis goes down, bring inflammation down, and we see neurogenesis go up. It's that simple and that powerful. There's a great deal being written on this, both in the consumer media, like in the Time magazine, where we talk about inflammation, the role in not only the heart attacks and the cancer, but also in Alzheimer's and cognitive decline. And of course, there are practitioners who've been writing about this relationship between inflammation and the brain. This is just one of the books, The Inflamed Mind, An Approach to Depression. When we have patients, it's an absolute first thing that we have to deal with is there inflammation? Look at the inflammation panel, look at your biomarkers. Almost always it's got to be your first port of call. Okay, next one, toxic exposure. So this is Pizzorno. Pizzorno is the expert in, he knows more about toxins than anyone I've ever come across. This is just one fantastic article. Again, I have it if you want to read it. It's called The Polluted Brain. And it talks about all these kind of microparticles that we are susceptible to that are actually in the air, that are coming from pollution, that are coming from walking out cars, factories, neurotoxins. So just one of the quotes from Lancet was, the risk of dementia increases tenfold comparing those living within 50 meters of a major highway compared to those living at least 150 meters. That's only 100 meters difference, and you've got a tenfold increased risk in dementia. So Pizzorno talks about the worst neurotoxins, and he differentiates between those that are the worst prenatally when we're pregnant, and then once post-pregnancy, what are the ones that influence us the most. And he has an amazing talk on that literally goes into the detail of all of them. He talks a great deal about mercury. He shows, he talks a lot about arsenic and how prolific it is, that it's in our drinking water. Now remember, this is an American-centric conference I went to, so most of the statistics come out of the States. And interestingly, when you look at the map of distribution for Alzheimer's, MS, Parkinson's, all of those diseases, it's very much sitting in Northern America. And if you actually look at Africa, it's probably the lightest. So it doesn't mean that we're not immune, we certainly are, but there's something happening in North America that is making these particular conditions that much more prevalent. Of course, it'd be interesting to look at South Africa and not Africa and see if we mimic, because we seem to mimic a lot of other things that the States do. This is one of the fantastic diagrams that comes out of the article, where it shows how we're actually taking in. So it's not that we just have to drink water, plastic bottled water, but actually one of the most powerful ways, obviously, to get neurotoxins into our brain is just to sniff it through our noses. And how that's actually the most direct route to get across the blood-brain barrier and to be able to get these kind of neurotoxins into our brain. So a really fantastic diagram. This is another amazingly interesting topic that can be spoken about by itself for hours on end. Julie Etlin's going to be speaking about Alzheimer's in more detail and about ApoE specifically. But one of the really interesting things, and not trying to duplicate what Julie's going to be talking about, is this Alzheimer's really is based on plaques and tangles, which Julie will talk about more. And what is one of the most interesting things about Alzheimer's is when they have gone in and done brain biopsies of people who've died with Alzheimer's, and they've kind of opened up these plaques and tangles, what they found inside are viruses, bacteria, and yeasts and molds. And I'm going to show you a little bit. How about that? I just loved the title of this paper. I thought it was absolutely genius. I can never think of anything that interesting. But really what they're trying to say is that what this kind of Alzheimer's disease, plaques and tangles, is actually the immune defense network doing its job. So in other words, plaques are really being generated in response to infection in the brain. So when our yeasts and our bacteria and our viruses have crossed over the blood-brain barrier, our brain mounts an immune response to be able to shut it down, and the way it does this is by creating these plaques around it. And I'm going to show you some really interesting stuff, a great article. So Tansey is just an extraordinary scientist because he kind of invents these 4-dimensional Petri dish Alzheimer's in a dish, but he also wrote a book with Deepak Chopra. And he works with Deepak Chopra. So he's one of those extraordinary scientists who's able to use both the left and the right side of the brain and really get beyond just kind of phenotype. Really good book. It's Super Brain, Super Genes, I think it's called. Anyway, him and a colleague invented this Alzheimer's in a dish, for which he has won many, many awards. And basically what they did was they were saying, he was like, we can't do mouse studies to understand what happens in the human brain, because actually the human brain works differently from the mouse brain. And so they needed to come up with another way to be able to find out what are the variables that are influencing our brain function and how do we measure that input. And we can't ethically do it on humans. So they came up with this Alzheimer's in a dish, which literally is that. It's a Petri dish, which has got nerve cells from human stem cells. Put in this dish, and then they put in whatever it is. They'll put in mercury, or they'll put in viruses, or they'll put in bacteria, or they'll put in a mold, or they'll put in Borrelia, whatever it is, they'll put in a vector, and they'll watch what happens in the Petri dish. And then they develop this extraordinary 4D technology. So in normal gels that we run in a lab, we have a 2D technology. You run the gel and you kind of look at it. They developed a 4-dimensional gel, and I actually don't have he presented a video where you can actually go and turn this around, and you can see exactly how the plaques, the nerve cells are reacting, the plaques are being built. It's just extraordinary technology. So he developed this Petri dish, but also this 4-dimensional. And this is one of the most interesting things that he found, is that the beta-amyloid was binding and trapping microbes. And this is one of the pictures. On the outside, we're seeing the beta-amyloid, and on the inside, we're actually seeing the microbes. That we had this kind of microbe infestation, and then you saw that the beta-amyloid just came and surrounded it and almost encapsulated it. I don't know if you can see this. You can't see this really clearly. But this is using the Alzheimer's in a dish technology. And what they found was that there was a bacterial brain infection, which triggered the plaque exactly. This was done in mice. And if you look at the bottom, which you can't see so clearly, unfortunately, but if you look at the bottom, there's little red dots there. What they did was they introduced salmonella into the, so this wasn't the Petri dish, my apologies. They introduced salmonella into the mouse, okay, infected it, and then they watched what happened. And the green really are the plaques. So in direct response to the introduction of salmonella in the brain, the plaques surrounded the salmonella, and if you could look here, you'd be able to see that there's little red dots in the middle of the green to show how they surround them and shut them down. Okay, end of infection, as much as I'd love to carry on talking about it. Oh, I did miss something. One of the most common viruses that is identified in this kind of brain, this infection hypothesis, is herpes simplex, by far. And a lot of the research is actually, and which is so common. So quite frightening. When I was doing my research for this talk, I didn't know whether to laugh or cry. I was like feeling like, oh my God, I'm going to go live on another planet where there's no neurotoxins and there's no, I actually didn't, and just walk all day. Okay, gut-brain connection.
Away from roads.
Away from roads, yeah. Walk, I don't know where. When we were in Johannesburg doing the estrogen workshop with Craig Golding. So we were in Johannesburg and we did an estrogen workshop with Dr. Craig Golding, a functional medicine practitioner, and he made a great commentary. We were talking about exposure to estrogens in the environment. He said, short of moving to another planet, and that was pretty it. It's like, where do we go? You know, we're just surrounded by this toxic environment. And then I was watching Handmaid's Tale, and then it was worse, because I was like, oh, this is what happens if we keep on polluting our planet. We're all going to become infertile. Anyway, so, you know, let's be more positive. Gut-brain connection. So this is something that I'm preaching to the converted. And my colleague, Dr. Anna Noy, spoke on this all the time. and in fact she used to talk about gut-brain immune, which is completely correct, that there is a direct connection between how our brain is functioning and our gut. The other thing we spoke about a great deal in Johannesburg was about phase 3 detoxification, which is obviously the gut. Leaky gut, gut permeability, toxins being reabsorbed through the gut back into the system. The founder of, not the founder, because it's probably a lot of people, but one of the most outspoken people in the space has always been Dr. Perlmutter. He wrote one of the first books, which I'll talk about, but this is one of the slides that actually Anne showed us, which is that when there's a trauma to the brain, and that trauma can be both physical, as in concussion, a car accident, violence, abuse, or it can be emotional trauma. It always manifests in the gut. And this really just takes the pathway of how do we go from trauma, which of course is around neuroinflammation, breakdown of the blood-brain barrier, dysregulation of cortisol, and then how do we land up with this kind of loss of brain integration, disruption of the brainstem integration, and right at the bottom we land up with the gastrointestinal dysfunction. Put that together with a kind of a dysbiotic gut, less than ideal diet and lifestyle, and it's very hard for us to be able to address cognitive function, albeit Alzheimer's or memory loss, albeit depression, anxiety, or mood disorders, when we have a dysbiotic gut. They are intrinsically linked. This was the first book he wrote. He has written many, many books since then. He's an amazing presenter to engage. He's probably the best presenter. He's always got beautiful videos in his presentations. And he was the first to say, if we're going to address the brain, we need to remove wheat, sugar, carbs in general. So he takes everything out. He takes out all grains, actually. And definitely goes very low carb and obviously no sugar. And he has since written cookbooks and he's quite prolific. But he was the first one to really, in the kind of public domain, talk about this idea that the food we eat impacts the way our brain works. So obviously the gut-brain connection, and that's our space, right? That's where we work. And that's one of the most powerful places for us to work. Okay, my space, genetics. So it's going to be frighteningly brief. So how did I address it? So clearly it's important. Third leading cause, 50 million people in the world with Alzheimer's, not to mention those that are hitting on the trajectory. I think one of the most, before I get into the genetics, one of the things that I should have said and haven't said is that why are there no cures and why do we not know Alzheimer's survivors? The answer is because patients do not present until they have the disease. So what is different with cancer and heart disease is we have all these processes in place that Vitality and Discovery pay for to be able to do early detection. So we have biomarkers, we have blood chemistry, we have mammograms, we have all these different ways to say, if we identify cancer early, we can intervene. If we identify heart disease early, because we know that chronic disease occurs over decades. Cognitive decline is no different. It's a chronic disease that happens in that space. And yet, when do patients present with cognitive decline and Alzheimer's? When they have it. And then there's very little we can do. We can delay, partially reverse, but we can't have these Alzheimer's survivors. So the most important thing is to understand what is the interventions and say we should be pushing to be able to identify 20 years before the patients that are at most risk of cognitive decline, mood disorders, anxiety, Alzheimer's, MS, whatever it is, and start intervening now. So that is the most important thing I actually want to share today, and it's a single thing I forgot to say. Okay, so what is genetics in this space? Well, genetics is a biomarker. Genetics is one of the insights that we can gather 20, 30 years before the plaques start forming in our patients' brains. Okay, or any of the other conditions that we're talking about, anxiety, depression, mood disorders. Okay, so it had to be inside the report. It couldn't be on the outside of the report. This isn't like, should we test for brain? Is brain important? Is cognitive function important for this individual? Well, they're not suffering from anything right now, so we don't need to test it. Absolutely not. Remember the endemic nature of cognitive decline. every patient who walks into our practice is either in the 10 or the 20 or the 30 years, or the beginning of it or the end of it. We need to know, are there any genes that are impacting metabolic processes or biochemical processes that would contribute to an increased decrease of, or an increased cognitive decline? Okay. We want to know now. It's one of the insights. It's one of the biomarkers. So in 3X4, we have addressed it by making it one of our pathways. We have put in a number of genes which cover some of the information around a brain, whether it's methylation, detoxification, BDNF, oxidative stress obviously is a really big one, memory loss. So we have built brain health. And as I said, I never ever look at brain health actually in isolation of either detoxification, inflammation, but actually in cellular systems, I pretty much look at all of these except for probably, well, actually even blood clotting I'd look at. So these are the most important, because one of the things we're going to talk about when it comes to intervention is that hormones play a fundamental role in managing cognitive health or brain health. So we want to know what's happening in terms of hormone balance and hormone metabolism. Glucose and insulin, they talk about Alzheimer's as this kind of other diabetes of the brain. It's almost always got an insulin-resistant phenotype associated with it. So we always have to consider insulin resistance. Vascular health is obviously huge. We also talk about Alzheimer's and cognitive decline as being a vascular disease, because that's essentially what's happening is we're shutting down the vessels in the brain. Blood clotting, brain health by itself, DNA damage, methylation, inflammation, oxidative stress, and detox. Of course, if we're taking in neurotoxins, if we're taking in all these things, how are we managing them? If we're taking in hormones, how are we managing them? How do we react to the exposure to external estrogens, to external toxins? So even though brain health is one pathway, it is not the only way to address brain health. And we have to consider all our cellular and all our system pathways. Okay, and then these, I've just taken the four that most important for me that from the page, but as I said, all the ones before important. So the ones that we put into brain health, some of those obviously a lot around dopamines. I didn't mention that, our neurotransmitters, dopamine behavior, dopamine metabolism, bless you. And you can kind of see oxidative stress, methylation, and then obviously we've got vascular health, which brings a lot of them together, lipid metabolism, detox, and inflammation. So just two patients quickly, one of which is me. So this patient has a really difficult presentation, suffered from a lot of history of gut dysbiosis, gut issues, all the kind of the worst kind of manifestations of a problematic gut right through childhood and into adulthood. Suffered a lot of depression, anxiety, was diagnosed with an anxiety disorder, was put onto medication. The medication worked in the beginning and then stopped working. Has a history of abuse in childhood by a parent. So we're already seeing a picture of dysbiotic gut. So remember, gut-brain connection. We are seeing abuse, which always has a function at a cognitive level that kind of remember the gut-brain connection. And we look at the genes, and we're seeing that the phase 2 detoxification is severely impacted. You can see, I mean, a double deletion, methylation, TT homozygote, and NQO1. I mean, that's significant. Okay, so in this patient, I mean, what we really would have liked to have done is intervene really, really early on, obviously, a gut protocol and improving gut. This is actually one of Annalie's patients, so she knows better than I, but this is exactly what she would be doing, is a gut protocol, and then obviously a detox protocol to be able to upregulate phase 2 detoxification, and then seeing how all of this impacts, and just those two I've picked out, but really how does all of this impact on the anxiety and mood disorder, because I think we will see a significant difference in this kind of patient. The reason that I have included my own, that's not mine, that's me. Oh, sorry, that's his. So you can see right at the top, we've got detoxification, DNA damage, inflammation, methylation, all the ones that we were concerned about. Brain health has come up, vascular health, and again, we've got a hormone balance. And actually, part of his medical history was he had very low testosterone, and he also had a real problem with cortisol. So cortisol was low in the day, in the morning, and very high at night. Major sleep challenges. So you can see the picture coming together, and it's a perfect case study for what we're talking about in cognitive health. Okay, well, the reason I want to show you this is because you can see that brain health has come up high, and there's no ApoE, ApoE33. So not an issue, not a drama, but brain health is still high. Then I'll show you this report where we've got an ApoE34, we've got an MTHFR-TT, and this is me. And yet, so it was interesting. I have always known that I had MTHFR-TT and ApoE34, and I have a family history of Alzheimer's. My father has 3 sisters, 2 out of 3 got Alzheimer's, 1 very young. And so I have lived in this Alzheimer's space a long time, and have always carried that fear around with me, and have only ever really known my genes, and assuming that I had 3, 4, and I was like, carried a fear, as we do when we have a strong family history. And the interesting thing we're seeing now is that the fear of Alzheimer's is far greater than the fear of cancer. It's far greater. And when people are having to choose, should I rather prevent Alzheimer's or prevent cancer, they're choosing, I'd rather prevent Alzheimer's every single time. This is like the shark in the water, right? It's taken over from cancer. So I always had this kind of sense of like, this is my path, and what am I going to do? And I've had this conversation with Julie many times, like, what am I going to do? Because I really don't want, I'm terrified of getting Alzheimer's. And then I got my report. Fantastic. I was like, it's, well, quite perfect, right? But you can see why I got carb issues. Yeah. So my carb issues are clearly there. But anyway, but I was like, wow, you're kidding me. Here I've been carrying around this genetic kind of thing, thinking that's my trajectory. But actually, that is amazing. And I had taken on, because I was only looking at MTHFR and only looking at ApoE34, I had kind of assigned this major heaviness and seriousness to that. But actually, when it was put into the context of everything else I had, I was like, hey, I'm in with a really good chance here. I really, if I make the right diet choices, make the right exercise choices, try not to overwork too much, sort of see, and try not to get too many of these neurotoxins into my body, I actually stand a real chance of not getting Alzheimer's when I'm older and keep learning things, which hopefully I'll keep doing. So for me, it was an absolute aha moment. Even though I built the test, I was so surprised to see my report. It was not at all what I expected. I absolutely expected to have a purple in my brain health. So it changed my whole process of thinking. I was like, oh, okay, so maybe ApoE isn't this absolute kind of sentence, and it isn't. And Julie will talk a little bit more about it. Okay, so I've really identified for you what are the most important areas, and you can see it's an incredibly exciting space. Any one of them by himself can be understood and unpacked a great deal more, and they did for 3 days. But let's have like 3 slides on how to treat this because we don't have time today. But I'm going to tell a little bit more about what were the things that were the most important variables that were identified in terms of treating brain health and cognitive function. And literally it's 3 slides, so you'll forgive me for that, but we can have conversations at another time going more in depth into what they are. But before we go into that, I just want to talk briefly about this concept of a roof with 36 holes, and apologies if we like crossing over, but Dale Bresden has written many books on, he's an Alzheimer's specialist really, but now he's moving into kind of MS and Parkinson's, and he actually has a residential clinic. So you go and you stay there and you get worked on, and he teaches a lot, and Julie's been done his courses. Is that he came with this idea of 36 holes, and what it means is there's no one way to treat cognitive decline. There's not one solution, and there is no drug. There is no drug that's ever succeeded in impacting it. They just manage symptoms of drugs. And one of the problems with the way we've been trying to treat Alzheimer's, especially with medication, is this idea of monotherapy. That if we give a drug and measure its effect, we have a solution. We do not have a solution. And he came up with this idea of 36, a roof with 36 holes. There are probably 36 things, more or less, that we need to do with our patients to be able to reverse cognitive decline. And only one of those is medication. And all the speakers said, if medication was measured together with all the other interventions, you would have seen that the medication actually played a role in reversing a thing. But by itself, it was completely ineffective. So this is a very functional medicine approach, which all of us here kind of appreciate and understand, and yet mainstream medicine hasn't kind of jumped on the bandwagon. So let's have a look at what those other 35 may be. So just to put it into perspective, the most important thing we're trying to understand here is if we want to prevent cognitive decline, and we don't want people to present when they're already fully asymptomatic, one is we need early prediction. What are the biomarkers? Genetics is only one of them. What are the other biomarkers? What are the other tests? What are the other that we can use to be able to identify 20 years prior? Prediction is more genetics. Detection is almost what can we actually measure? BDNF is an example. And then early intervention. Early and lifetime intervention. We need to intervene before we have symptoms. This is the slide. Everything in one slide. Okay. And yes, you can have the slide if you want it. If you want it. So you can imagine how much goes into each block. But really, exercise, exercise, exercise. 50% of everything we do is exercise. So if we have a basket of tools, the most powerful tool in our basket is 50%. The other 50% is everything else that's in the picture. By far, exercise is the trump card of all. And one of the problems they have is when you experience such severe cognitive decline, dementia, Alzheimer's, we're at the point where you dissociate or lose self-awareness. It's extremely difficult to get patients to exercise, extremely difficult. And that's why it needs to be happening so much earlier. Because once they have passed a certain point, it's very hard for all of these interventions, diet, nutrients, exercise. Okay, so exercise, trump card by far. Okay, so I was very happy to have a physio in our audience today. Healthy diet. So, we put the whole of diet into one little square. So what are they talking about? They're talking about lower carb, no refined carbohydrates, no sugar, kind of like paleo-Mediterranean is really what they're looking at. Healthy fats, and then of course, toxin-free is one of the most important. So a clean food. So there's obviously a lot of detail. And then when we are very symptomatic, they obviously use the ketogenic diet, MCT ketogenic diet. That's when we're presenting with a lot of symptoms. But in the pre-symptom time, they talk about obviously clearing out all the refined food, refined carbohydrate, and obviously sugars being an absolute no. And healthy fats, lots of fruit and vegetables. Terry Stolz, I don't know if you know Terry Stolz, who had MS. She presented at IFM as well, and she talks about her kind of 9 cups of, she kind of follows a modified paleo type diet, 9 cups of vegetables a day, lots of different colors, and some of the other things that she does. And obviously wild fish and no hormones in our meat, so she does use meat and fish, is a major part, going back to what Pizzorno talks about in terms of toxins. Okay. Nutrients, I actually have another slide, and the slide is actually all the nutrients. So all the speakers recommended 1 or 2 things in terms of nutraceuticals and nutrients, and I've actually put it together in one slide for you, which I'll show you. The ones that have been shown in the Alzheimer's in a dish and in trials to really improve brain function. Social interaction comes through hugely. Some of the entire talks were just about social interaction, about connection. When you're isolated and you don't have human connection, cognitive decline increases significantly. And of course, as you become a greater cognitive decline, that connection gets harder and harder. And again, why it needs to happen so earlier. Learning new things, love this one. A great deal was spoken about this, about why does it work. Obviously, what we want to do is it's exactly like exercise. If we don't use it, we lose it. If we want to grow more muscle, we have to train more. It's exactly the same with the brain. If we want to have neurogenesis happening, we need to utilize the brain. There's one of the talks was about Brain HQ, which is an online computer program, really fascinating. That helps. There's a couple of them in the marketplace. This is one of the talks about what kind of learning does it need to be, and when should we start it. And it's not just for Alzheimer's. We're talking about ADHD, ADD, behavioral issues in children. It is extremely powerful and can be started very early, and can be on an iPad. And there's, it's not just a case of learning a new language. There's really some science behind it, and being able to measure where you are. So any of us can go into Brain HQ, go in, register, and be able to assess where we are in our, almost like being able to put these puzzles together. And the great thing about Brain HQ is it actually will, say some of us, most of us have very high cognitive function. It'll set us at a very high bar and just keep on going. It's a fascinating program, really, really good for children, although there are other programs for kids as well. So learning new things. We have to, one of the greatest comments I heard is that sitting is the new smoking. Sorry, that was for you, Jax. Sitting is the new smoking. I just thought that was absolutely powerful. What do? I sit 8 hours a day. I literally sit 8 hours a day. It was such a wake-up call for me. Adequate sleep. What does sleep do? Amongst many things, sleep clears the plaques from our brain. That's what happens while we're sleeping. It's when we clear, but it has to be deep sleep. It has to be quality sleep to be able to clear those plaques from our brain. If we're not sleeping deeply and we're not getting enough sleep, we are increasing our risk for cognitive decline significantly. Again, there's so much literature behind this, lots and lots of literature. Stress reduction, meditation, mindfulness, absolutely imperative in terms of cognitive function and optimizing cognitive. Meditation is not sitting out there in Nurtuk and Kormaki anymore. Meditation is a mainstream intervention for our patients. And in cognitive function, it is absolutely fundamental. Appropriate hormones. We have some hormone specialists in our audience today. And there's been, again, I think 2 of the talks were entirely about hormones, and this idea that what was really interesting to me is that the use of, they all talk about bioidentical hormones and how you can use them to be able to manage cognitive decline, reverse a lot of symptoms, and delay. And one of the really interesting discussions was around this idea that if you have a patient, and even if you're looking at them genetically or family history or biomarker, and they have a risk for cognitive decline, or they're showing that they move in that direction, and they may have a risk for breast cancer later in life, and you need to make decisions around that. You need to understand what you're doing, because, as I said, there's some people who are going to say, "Give me the hormones. I'm going to take my chances on breast cancer, and I'm going to go, but I want the protection." So it's a really interesting space, and there's a lot of work being done about how powerful hormones can be in reversing, delaying, and managing cognitive decline. So I don't know very much about it, but it was very interesting because I'd always only thought about hormones through the lens of cancer. That was my lens. And this opened up a whole new world for me. Toxin reduction. How do we in our lives on every level, water, air, food, exposures, reduce our toxic exposure, and how so important it is in terms of this kind of maintaining brain neurogenesis and brain integrity. And medication. So they're medications that are going to help us, and they're medications that are going to harm us. And obviously antibiotics comes back to the gut-brain connection. Every course of antibiotics, every course of antibiotics changes your microbiome forever. You never get back to exactly where you were when you take the antibiotics. So just understanding how that connection affects brain, not just microbiome. And my last slide, these are the ones that came up in all the talks. Some really, some that you're probably extremely familiar with, some that I wasn't familiar with. DHA and EPA, obviously inflammation, we understand that. Cat's claw, ashwagandha, no surprise at all. It's finding its way again into kind of mainstream clinicians, which is wonderful. The one I didn't know is nicotinamide riboside. Is anyone here familiar with it? I don't know if it's available in South Africa. It comes under a name called Niagen, I think. I think there's one company in the world that owns the patent, and they sell it under different brands. And I was very interested to see if we can get some of our reps onto it and see if anyone knows if we can get it in the country. Skull cap, I contacted Carla Boswell. Unfortunately, Carla's not here today. She's out the country because I was like, I want some of that. I want some of that. So when they put it into, so there's two types of skull cap apparently, which is what Carla explained to me. The one they talk about in the presentations is Bicodin. I could get it wrong. And when they put it into the Alzheimer's in a dish, Petri dish, they saw incredible reversal of plaques and tangles. Carla says that both are available. Both skull caps are actually very useful. One more at a mood level, and the other one at a kind of a neuroinflammation level.
Available at
Phyto Force in the drop form. The skull cap. Don't know which one, but maybe it would be interesting to find out. Okay, great. Coffee is back in. Coffee is back in. Okay. All the studies were done on the whole coffee fruit, not on the cappuccino. But definitely, so this is obviously the conversation is around, what's the difference between having an espresso and utilizing this whole coffee fruit. But the research around whole coffee fruit has been extraordinary. And in fact, caffeine per se is incredibly powerful in terms of neurogenesis. And obviously understanding again, how we use it, when we use it, if we metabolize it really fast or really slow, how do we use it with our patients? But also what I didn't get to understand and needs more research is the difference between using coffee that we recreationally drink versus using the whole coffee fruit as a supplement, which apparently is extremely powerful. Have you come across it? Coffee fruit extract, yeah.
Not the green coffee bean.
No, I don't think so. It's the whole coffee fruit before they take the bean out, is my understanding. Am I right? It's actually the coffee fruit, which they then open and extract the bean they make coffee with. So it's actually the fruit that we're after. Turmeric and curcumin, you all know that one, for its inflammatory processes. And then alpha-lipoic acid, an extremely, extremely important vitamin D. Vitamin D is like the no-brainer, if we don't have adequate vitamin D levels, no brainer. Oh, that was very good. Thank you. Where's Jason? I got a pun. Jason's like our pun king, so that was. I didn't even notice when I made it. So vitamin D, we cannot achieve brain health on low vitamin D levels. It's, as you say, the no-brainer. My last slide. I love this slide. They did. This was published in Nature. So this version wasn't, but they wrote an editorial in Nature, which I love. And you can see Tansy's there again, where they went and did a full study on the impact of going on holiday, on vacation, on disease-associated molecular phenotypes, including brain function. And then they measured to see whether brain function improved and BDNF went up and all of this great stuff if you went on holiday. And then they said, what happens if you go on holiday and meditate while you're on holiday? And the results just went through the roof, if you'll excuse the pun, 36. Anyway, so it was an incredible, powerful study with an amazing, basically saying that, I love the end, for those already trained in the practice of meditation, a retreat appears to provide additional benefits to cellular health beyond the vacation effect. Go on holiday, meditate, best outcome you can have in terms of brain function. And that's why we all need to make sure we're not overworking, we're taking leave, we're taking time out, and we're getting away. I know some of us are better than others. That's me for today. Thank you.